Introns range in length from a single nucleotide to more than 10,000 nucleotides. Some eukaryotic genes lack introns altogether. Some have only a few, but most have many. Figure 718. Introns are removed by RNA splicing. To produce an mRNA in a eukaryotic cell, the entire length of the gene, introns as well as exons, is transcribed into RNA. After capping, and as the RNA polymerase continues to transcribe the gene, the process of RNA splicing begins, in which the intron sequences are removed from the newly synthesized RNA and the exons are stitched together. Each transcript ultimately receives a poly-A tail. In some cases, this happens after the splicing, and in other cases it occurs before the final splicing reactions have been completed. Once a transcript has been spliced and its 5' and 3' ends have been modified, the RNA is a functional mRNA molecule that can now leave the nucleus and be translated into protein. How does a cell determine which parts of the RNA transcript to remove during splicing? Unlike the coding sequence of an exon, most of the nucleotide sequence of an intron is unimportant. Although there is little overall resemblance between the nucleotide sequences of different introns, each intron contains a few short nucleotide sequences that act as cues for its removal. These sequences are found at or near each end of the intron and are the same or very similar in all introns. Figure 719. Guided by these sequences, an elaborate splicing machine cuts out the intron sequence in the form of a lariat structure, figure 720, formed by the reaction of the A highlighted in red in figures 719 and 720. Although we will not describe the splicing machinery in detail, we note that, unlike the other steps of mRNA production we have discussed, RNA splicing is carried out largely by RNA molecules instead of proteins. RNA molecules recognize intron-exon boundaries through complementary base pairing and participate intimately in the chemistry of splicing. These RNA molecules, called small nuclear RNAs, snRNAs, are packaged with additional proteins to form small nuclear ribonucleoprotein particles. snRNPs, pronounced SNRPs. The SNRPs form the core of the spliceosome, the large assembly of RNA and protein molecules that carries out RNA splicing in the cell. To watch the spliceosome in action, see movie 7.5. The intron-exon type of gene arrangement in eukaryotes at first seems wasteful, but it does have positive consequences. First, the transcripts of many eukaryotic genes can be spliced in different ways, each of which can produce a distinct protein. Such alternative splicing thereby allows many different proteins to be produced from the same gene. Figure 721. An estimated 60% of human genes probably undergo alternative splicing. Thus, RNA splicing enables eukaryotes to increase the already enormous coding potential of their genomes. RNA splicing also provides another advantage to eukaryotes, one that is likely to have been profoundly important in the early evolutionary history of genes. As we discuss in detail in Chapter 9, the intron-exon structure of genes is thought to have speeded up the emergence of new and useful proteins. The inclusion of long introns makes genetic recombination between exons of different genes much more likely. This means that genes for new proteins could have evolved quite rapidly by the combination of parts of pre-existing genes, a mechanism resembling the assembly of a new type of machine from a kit of pre-existing functional components. Indeed, many proteins in present-day cells resemble patchwork composed from a common set of protein pieces, called protein domains. See figure 416. Mature eukaryotic mRNAs are selectively exported from the nucleus. We have seen how eukaryotic mRNA synthesis and processing takes place in an orderly fashion within the cell nucleus. However, these events create a special problem for eukaryotic cells. Of the total mRNA that is synthesized, only a small fraction, the mature mRNA, is useful to the cell. The remaining RNA fragments, excised introns, broken RNAs, and aberrantly spliced transcripts are not only useless but could be dangerous to the cell if not destroyed. How, then, does the cell distinguish between the relatively rare mature mRNA molecules it needs to keep and the overwhelming amount of debris generated by RNA processing? The answer is that the transport of mRNA from the nucleus to the cytoplasm, where it is translated into protein, is highly selective. Only correctly processed RNAs are allowed to pass. This reliance on proper processing for RNA transport is mediated by the nuclear pore complex, which recognizes and exports only completed mRNAs. These aqueous pores connect the nucleoplasm with the cytosol, and as we discuss in Chapter 15, they act as gates that control which macromolecules can enter or leave the nucleus. To be export ready, an mRNA molecule must be bound to an appropriate set of proteins, each of which signals that the mRNA has been correctly processed. These proteins include poly-A binding proteins, a cap binding complex, and proteins that mark completed RNA splices. Figure 722. It is presumably the entire set of bound proteins rather than any single protein that ultimately determines whether an RNA molecule will leave the nucleus. The waste RNAs that remain behind in the nucleus are degraded, and the building blocks are reused for transcription. mRNA molecules are eventually degraded by the cell. Because a single mRNA molecule can be translated many times, see Figure 7.2, the length of time that a mature mRNA molecule persists in the cell affects the amount of protein it produces. Each mRNA molecule is eventually degraded into nucleotides by RNases, but the lifetimes of mRNA molecules differ considerably, depending on the nucleotide sequence of the mRNA and the type of cell in which the mRNA is produced. Most mRNAs produced in bacteria are degraded rapidly, having a typical lifetime of about 3 minutes. The mRNAs in eukaryotic cells usually persist for longer. Some transcripts, such as that encoding beta-globin, have lifetimes of more than 10 hours, whereas other eukaryotic mRNAs have lifetimes of less than 30 minutes. These different lifetimes are in part controlled by nucleotide sequences that are present in the mRNA itself, most often in the portion of RNA called the 3' untranslated region that lies between the 3' end of the coding sequence and the poly-A tail. The different lifetimes of mRNA help the cell specify the amount of each protein that it synthesizes. In general, proteins made at high levels, such as beta-globin, are translated from mRNAs that have long lifetimes, whereas those proteins present at low levels, or those whose levels must change rapidly in response to signals, are typically synthesized from short-lived mRNAs. These different lifetimes are the outcome of evolutionary fine-tuning, in which the stability of each mRNA is tied to the needs of the cell. The earliest cells may have had introns in their genes. The process of transcription is universal. All cells use RNA polymerase coupled with complementary base pairing to synthesize RNA from DNA. Indeed, bacterial and eukaryotic RNA polymerases are almost identical in overall structure and clearly evolved from a shared ancestral polymerase. It may therefore seem puzzling that the resulting transcript is handled very differently in eukaryotes and in prokaryotes. Figure 723. In particular, RNA splicing seems to mark a fundamental difference between those two types of cells. But how did this dramatic difference arise? As we have seen, RNA splicing provides eukaryotes with the ability to produce a variety of proteins from a single gene, and grants them a certain degree of evolutionary flexibility. However, these advantages come with a cost. The cell has to maintain a larger genome and has to throw out a large fraction of the RNA it synthesizes. According to one school of thought, early cells, the common ancestors of prokaryotes and eukaryotes, contained introns that were lost in prokaryotes during subsequent evolution. By shedding their introns and adopting a smaller, more streamlined genome, prokaryotes would have been able to reproduce more rapidly and efficiently. Consistent with this idea, simple eukaryotes that reproduce rapidly, some yeasts, for example, have relatively few introns, and these introns are usually much shorter than those found in higher eukaryotes. On the other hand, some argue that introns were originally parasitic mobile genetic elements, discussed in Chapter 6, that happened to invade an early eukaryotic ancestor, colonizing its genome. These host cells then unwittingly replicated the selfish nucleotide snippets along with their own DNA, and modern eukaryotes have never bothered to sweep away the genetic clutter left from that ancient infection. The issue, however, is far from settled. Whether introns evolved early and were lost in prokaryotes, or evolved later in eukaryotes is still a topic of scientific debate, and we return to it in Chapter 9. From RNA to protein. By the end of the 1950s, biologists had demonstrated that the information encoded in DNA is copied first into RNA and then into protein. The debate then shifted to the coding problem. How is the information in a linear sequence of nucleotides in RNA translated into the linear sequence of a chemically quite different set of subunits, the amino acids in proteins? The fascinating question stimulated much excitement among scientists at the time. Here was a cryptogram set up by nature that, after more than 3 billion years of evolution, could finally be solved by one of the products of evolution, human beings. Indeed, scientists have not only cracked the code but have revealed, in atomic detail, the precise workings of the machinery by which cells read this code. An mRNA sequence is decoded in sets of 3 nucleotides. Transcription as a means of information transfer is simple to understand. Since DNA and RNA are chemically and structurally similar, and DNA can act as a direct template for the synthesis of RNA through complementary base pairing. As the term transcription signifies, it is as if a message written out by hand were being converted, say, into a typewritten text. The language itself and the form of the message do not change, and the symbols used are closely related. In contrast, the conversion of the information in RNA into protein represents a translation of the information into another language that uses quite different symbols. Because there are only 4 different nucleotides in mRNA, but 20 different types of amino acids in a protein, this translation cannot be accounted for by a direct one-to-one correspondence between a nucleotide in RNA and an amino acid in protein. The rules by which the nucleotide sequence of a gene, through the medium of mRNA, is translated into the amino acid sequence of a protein are known as the genetic code. The sequence of nucleotides in the mRNA molecule is read consecutively in groups of 3. Because RNA is a linear polymer made of 4 different nucleotides, there are thus 4 times 4 times 4 equals 64 possible combinations of 3 nucleotides, AAA, AUA, AUG, and so on. However, only 20 different amino acids are commonly found in proteins. Either some nucleotide triplets are never used, or the code is redundant, and some amino acids are specified by more than one triplet. The second possibility is in fact correct, as shown by the completely deciphered genetic code in Figure 724. Each group of 3 consecutive nucleotides in RNA is called a codon, and each specifies one amino acid. The strategy by which this code was cracked is described in How We Know, pages 248 to 249. The genetic code shown in Figure 724 is used in nearly all present-day organisms. Although a few slight differences in the code have been found, these occur chiefly in the DNA of mitochondria and of some fungi and protozoa. Mitochondria have their own transcription and protein synthesis machinery that operate quite independently from those of the rest of the cell, discussed in Chapter 14, and they have been able to accommodate minor changes to the otherwise universal code. Even in fungi and protozoa, the similarities in the code far outweigh the differences. In principle, an RNA sequence can be translated in any one of three different reading frames, depending on where the decoding process begins. Figure 725. However, only one of the three possible reading frames in an mRNA specifies the correct protein. We shall see in a later section how a special punctuation signal at the beginning of each RNA message sets the correct reading frame. tRNA molecules match amino acids to codons in mRNA. The codons in an mRNA molecule do not directly recognize the amino acids they specify. The group of three nucleotides does not, for example, bind directly to the amino acid. Rather, the translation of mRNA into a protein depends on adapter molecules that can recognize and bind to a codon at one site on their surface and to an amino acid at another site. These adapters consist of a set of small RNA molecules known as transfer RNAs, tRNAs, each about 80 nucleotides in length. We saw earlier that an RNA molecule will generally fold into a three-dimensional structure by forming base pairs between different regions of the molecule. If the base paired regions are sufficiently extensive, they will form a double helical structure, like that of double-stranded DNA. The tRNA molecule provides a striking example of this. 4 short segments of the folded tRNA are double helical, producing a molecule that looks like a clover leaf when drawn schematically. Figure 728a. For example, a 5' GCUC3' sequence in one part of a polynucleotide chain can form a relatively strong association with a 5' GAGC3' sequence in another region of the same molecule. The clover leaf undergoes further folding to form a compact L-shaped structure that is held together by additional hydrogen bonds between different regions of the molecule. Figure 728b and c. 2 regions of unpaired nucleotides situated at either end of the L-shaped molecule are crucial to the function of tRNA in protein synthesis. One of these regions forms the anticodon, a set of 3 consecutive nucleotides that through base pairing bind the complementary codon in an mRNA molecule. The other is a short single-stranded region at the 3' end of the molecule. This is the site where the amino acid that matches the codon is attached to the tRNA. We saw in the previous section that the genetic code is redundant. That is, several different codons can specify a single amino acid. See Figure 724. This redundancy implies either that there is more than one tRNA for many of the amino acids, or that some tRNA molecules can base pair with more than one codon. In fact, both situations occur. Some amino acids have more than one tRNA, and some tRNAs are constructed so that they require accurate base pairing only at the first 2 positions of the codon and can tolerate a mismatch or wobble at the 3rd position. This wobble base pairing explains why so many of the alternative codons for an amino acid differ only in their 3rd nucleotide. See Figure 724. Wobble base pairings make it possible to fit the 20 amino acids to their 61 codons with as few as 31 kinds of tRNA molecules. The exact number of different kinds of tRNAs, however, differs from one species to the next. For example, humans have nearly 500 different tRNA genes, but only 48 anticodons are represented among them. Specific enzymes couple tRNAs to the correct amino acid. We have seen that in order to read the genetic code in DNA, cells make many different tRNAs. We now must consider how each tRNA molecule becomes charged, linked to the one amino acid in 20 that is the right partner. Recognition and attachment of the correct amino acid depends on enzymes called amino-acyl tRNA synthetases, which covalently couple each amino acid to its appropriate set of tRNA molecules. In most organisms, there is a different synthetase enzyme for each amino acid. That is, there are 20 synthetases in all. One attaches glycine to all tRNAs that recognize codons for glycine. Another attaches phenylalanine to all tRNAs that recognize codons for phenylalanine, and so on. Specific nucleotides in both the anticodon and the amino acid accepting arm allow the correct tRNA to be recognized by the synthetase enzyme. Movie 7.6. The synthetases are equal in importance to the tRNAs in the decoding process, because it is combined action of synthetases and tRNAs that allows each codon in the mRNA molecule to associate with its proper amino acid. Figure 729. The synthetase catalyzed reaction that attaches the amino acid to the 3' end of the tRNA is one of many reactions coupled to the energy-releasing hydrolysis of ATP. See Figure 330, and it produces a high-energy bond between the charged tRNA and the amino acid. The energy of this bond is used at a later stage in protein synthesis to link the amino acid covalently to the growing polypeptide chain. The RNA message is decoded on ribosomes. The recognition of a codon by the anticodon on a tRNA molecule depends on the same type of complementary base pairing used in DNA replication and transcription. However, accurate and rapid translation of mRNA into protein requires a large molecular machine that moves along the mRNA, captures complementary tRNA molecules, holds them in position, and covalently links the amino acids that they carry so as to form a protein chain. This protein manufacturing machine is the ribosome, a large complex made from more than 50 different proteins, the ribosomal proteins, and several RNA molecules called ribosomal RNAs, rRNAs. A typical living cell contains millions of ribosomes in its cytoplasm. Figure 730. Eukaryotic and prokaryotic ribosomes are very similar in structure and in function. Both are composed of one large and one small subunit that fit together to form a complete ribosome with a mass of several million Daltons. Figure 731. For comparison, an average-sized protein has a mass of 30,000 Daltons. The small subunit matches the tRNAs to the codons of the mRNA, while the large subunit catalyzes the formation of the peptide bonds that covalently link the amino acids together in a polypeptide chain. The two subunits come together on an mRNA molecule, usually near its beginning 5' end, to begin the synthesis of a protein. The mRNA is then pulled through the ribosome like a long piece of tape. As the mRNA moves through it, the ribosome translates the nucleotide sequence into an amino acid sequence, one codon at a time, using the tRNAs as adapters. Thus, each amino acid is added in the correct sequence to the end of the growing polypeptide chain. Movie 7.7. Finally, the two subunits of the ribosome separate when synthesis of the protein is finished. Ribosomes operate with remarkable efficiency. In 1 second, a ribosome in a eukaryotic cell adds about 2 amino acids to a polypeptide chain. Bacterial ribosomes operate even faster, at a rate of about 20 amino acids per second. How does a ribosome choreograph all the movements required for translation? Each ribosome contains a binding site for an mRNA molecule and 3 binding sites for tRNA molecules, called the A site, the P site, and the E site. Figure 732. To add an amino acid to a growing peptide chain, the appropriate charge tRNA enters the A site by base pairing with the complementary codon on the mRNA molecule. Its amino acid is then linked to the peptide chain held by the tRNA in the neighboring P site. Next, the ribosome shifts, and the spent tRNA is moved to the E site before being ejected. Figure 733. This cycle of reactions is repeated each time an amino acid is added to the polypeptide chain, with the chain growing from its amino to its carboxyl end until a stop codon is encountered. The ribosome is a ribozyme. The ribosome is one of the largest and most complex structures in the cell, composed of 2/3 RNA and 1/3 protein. The determination of the entire 3-dimensional structure of its large and small subunits in 2000 was a major triumph for modern biology. The structure strongly confirms earlier evidence that the rRNAs, not the proteins, are responsible for the ribosome's overall structure and its ability to choreograph protein synthesis. The rRNAs are folded into highly compact, precise 3-dimensional structures that form the core of the ribosome. Figure 734. In marked contrast to the central positioning of the rRNA, the ribosomal proteins are generally located on the surface, where they fill the gaps and crevices of the folded RNA. The main role of the ribosomal protein seems to be to fold and stabilize the RNA core, while permitting the changes in rRNA conformation that are necessary for this RNA to catalyze efficient protein synthesis. Not only are the 3 tRNA binding sites, the A, P, and E sites on the ribosome formed primarily by the rRNAs, but the catalytic site for peptide bond formation is formed by the 23S RNA of the large subunit. The nearest amino acid is located too far away to make contact with the incoming amino acid tRNA or with the growing polypeptide chain. The catalytic site in this RNA-based peptidyl transferase is similar in many respects to that found in some protein enzymes. It is a highly structured pocket that precisely orients the two reactants, the elongating peptide and the charged tRNA, thereby greatly increasing the probability of a productive reaction. RNA molecules that possess catalytic activities are called ribozymes. In the final section of this chapter, we consider other ribozymes and discuss what the existence of RNA-based catalysis might mean for the early evolution of life on Earth. Here we need only note that there is good reason to suspect that RNA rather than protein molecules served as the first catalysts for living cells. If so, the ribosome, with its RNA core, could be viewed as a relic of an earlier time in life's history, when cells were run almost entirely by ribozymes. Codons in mRNA signal where to start and to stop protein synthesis. Although in the test tube, ribosomes can be forced to translate any RNA, see how we know pages 248 to 249. In the cell, a specific start signal is required. The site at which protein synthesis begins on an mRNA is crucial because it sets the reading frame for the whole length of the message. An error of one nucleotide either way at this stage will cause every subsequent codon in the message to be misread, resulting in a non-functional protein with a garbled sequence of amino acids. See Figure 727. The initiation step is also of great importance in another respect, because it is the last point at which the cell can decide whether the mRNA is to be translated. The rate of initiation thus determines the rate at which the protein is synthesized from the RNA. The translation of an mRNA begins with the codon AUG and a special tRNA is required to initiate translation. This initiator tRNA always carries the amino acid methionine, or a modified form of methionine, formal methionine in bacteria, so that newly made proteins all have methionine as the first amino acid at their N-terminal end, the end of a protein that is synthesized first. This methionine is usually removed later by a specific protease. The initiator tRNA is distinct from the tRNA that normally carries methionine. In eukaryotes, the initiator tRNA coupled to methionine is first loaded into a small ribosomal subunit, along with additional proteins called translation initiation factors. Figure 735. Of all the charged tRNAs in the cell, only the charged initiator tRNA is capable of binding tightly to the P site of the small ribosomal subunit. Next, the loaded ribosomal subunit binds to the 5' end of an mRNA molecule, which is signaled by the cap that is present on an eukaryotic mRNA. See Figure 716. The small ribosomal subunit then moves forward 5' to 3' along the mRNA, searching for the first AUG. When this AUG is encountered, several initiation factors dissociate from the small ribosomal subunit to make way for the large ribosomal subunit to assemble and complete the ribosome. Because the initiator tRNA is bound to the P site, protein synthesis is ready to begin with the addition of the next charged tRNA to the A site. See Figure 735. The mechanism for selecting a start codon is different in bacteria. Bacterial mRNAs have no 5' caps to tell the ribosome where to begin searching for the start of translation. Instead, they contain specific ribosome binding sequences, up to six nucleotides long, that are located a few nucleotides upstream of the AUGs at which translation is to begin. Unlike a eukaryotic ribosome, a prokaryotic ribosome can readily bind directly to a start codon that lies in the interior of an mRNA, as long as a ribosome binding site precedes it by several nucleotides. Such ribosome binding sequences are necessary in bacteria, as prokaryotic mRNAs are often polycistronic, that is, they encode several different proteins, each of which is translated from the same mRNA molecule. Figure 736. In contrast, a eukaryotic mRNA usually carries the information for a single protein. The end of the protein coding message in both prokaryotes and eukaryotes is signaled by the presence of one of several codons called stop codons. See Figure 726. These special codons, UAA, UAG, and UGA, are not recognized by a tRNA and do not specify an amino acid, but instead signal to the ribosome to stop translation. Proteins known as release factors bind to any stop codon that reaches the A site on the ribosome, and this binding alters the activity of the peptidyltransferase in the ribosome, causing it to catalyze the addition of a water molecule instead of an amino acid to the peptidyl tRNA. Figure 737. This reaction frees the carboxyl end of the polypeptide chain from its attachment to a tRNA molecule, and because this is the only attachment that holds the growing polypeptide to the ribosome, the completed protein chain is immediately released into the cytosol. The ribosome releases the mRNA and associates into its two separate subunits, which can then assemble on another mRNA molecule to begin a new round of protein synthesis. We saw in Chapter 4 that many proteins can fold into their three-dimensional shape spontaneously, and some do so as they are spun out of the ribosome. Most proteins, however, require molecular chaperones to help them fold correctly in the cell. The best studied chaperones use successive rounds of ATP hydrolysis to continually bind and release newly synthesized proteins until they are properly folded. This handling steers the protein along productive folding pathways by preventing them from forming aggregates or other misfolded structures. Newly synthesized proteins are typically met by their chaperones as they emerge from the ribosome. Proteins are made on polyribosomes. The synthesis of most protein molecules takes between 20 seconds and several minutes. But even during this very short period, multiple initiations usually take place on each mRNA molecule being translated. If the mRNA is being translated efficiently, a new ribosome hops onto the 5' end of the mRNA molecule almost as soon as a preceding ribosome has translated enough of the nucleotide sequence to move out of the way. The mRNA molecules being translated are therefore usually found in the form of polyribosomes, also known as polysomes, large cytoplasmic assemblies made up of several ribosomes spaced as close as 80 nucleotides apart along a single mRNA molecule. Figure 738. These multiple initiations mean that many more protein molecules can be made in a given time than would be possible if each had to be completed before the next could start. Both bacteria and eukaryotes make use of polysomes, but bacteria can speed up the rate of protein synthesis even further. Because bacterial mRNA does not need to be processed and is also physically accessible to ribosomes while it is being made, ribosomes will typically attach to the free end of a bacterial mRNA molecule and start translating it even before the transcription of that RNA is complete. These ribosomes follow closely behind the RNA polymerase as it moves along DNA. Inhibitors of prokaryotic protein synthesis are used as antibiotics. The ability to translate mRNAs accurately into proteins is a fundamental feature of all life on Earth. Although the ribosome and other molecules that carry out this enormous task are very similar among organisms, there are some subtle differences, as we have seen in the way that bacteria and eukaryotes synthesize proteins. Through a quirk of evolution, these differences form the basis of one of the most important advances in modern medicine. Many of our most effective antibiotics are compounds that act by inhibiting bacterial but not eukaryotic protein synthesis. Some of these drugs exploit the small structural and functional differences between bacterial and eukaryotic ribosomes, so that they interfere preferentially with bacterial protein synthesis. These compounds can thus be taken in high doses without being toxic to humans. Because different antibiotics bind to different regions of the bacterial ribosome, these drugs often inhibit different steps in protein synthesis. A few of the antibiotics of this kind are listed in Table 7.3. Many common antibiotics were first isolated from fungi. Fungi and bacteria often occupy the same ecological niches. To gain a competitive edge, fungi have evolved over time potent toxins that kill bacteria but are harmless to themselves. Because fungi and humans are both eukaryotes and are thus more closely related to one another than either is to bacteria, see Figure 129, we have been able to borrow these compounds to combat our own bacterial enemies. Carefully controlled protein breakdown helps regulate the amount of each protein in a cell. After a protein is released from the ribosome, it becomes subject to a number of controls by the cell. The number of copies of a protein in a cell depends, like the human population, not only on how quickly new individuals are made, but also on how long they survive. So the breakdown of proteins into their constituent amino acids by cells is a way of regulating the amount of particular protein present at a given time. Proteins vary enormously in their lifespan. Structural proteins that become part of a fairly permanent tissue, such as bone or muscle, may last for months or even years, whereas other proteins, such as metabolic enzymes and those that regulate the cycle of cell growth, mitosis, and division, discussed in Chapter 18, last only for days, hours, or even seconds. How does the cell control these lifetimes? Cells possess specialized pathways to enzymatically break proteins down into their constituent amino acids, a process termed proteolysis. The enzymes that degrade proteins first to short peptides and finally to individual amino acids are known collectively as proteases. Proteases act by cutting, hydrolyzing the peptide bonds between amino acids. See panel 2.5, pages 72 to 73. One function of proteolytic pathways is to rapidly degrade those proteins whose lifetimes must be short. Another is to recognize and eliminate proteins that are damaged or misfolded. Eliminating improperly folded proteins is critical for an organism, as neurodegenerative disorders such as Huntington's, Alzheimer's, and Creutzfeldt-Jakob diseases are caused by the aggregation of misfolded proteins. These protein aggregates can severely damage cells and tissues and even trigger cell death. Although most damaged proteins are broken down in the cytosol, important protein degradation pathways also operate in other compartments in eukaryotic cells, such as lysosomes, discussed in Chapter 15. In eukaryotic cells, most proteins are broken down by machines called proteasomes. A proteasome contains a central cylinder formed from proteases whose active sites face into an inner chamber. Each end of the cylinder is stoppered by a large protein complex formed from at least 10 different types of protein subunits. Figure 739. These protein stoppers bind the proteins destined for digestion, and then, using ATP hydrolysis to fuel their activity, feed the doomed molecules into the inner chamber of the cylinder, where the proteases chop the proteins into short peptides, which are then released from the end of the proteasome. Housing proteases inside this molecular chamber of destruction makes sense, as it prevents them from running rampant throughout the cell. How does the proteasome select which proteins in the cell should enter the cylinder and be degraded? Proteasomes act primarily on proteins that have been marked for destruction by the covalent attachment of a small protein called ubiquitin. Specialized enzymes tag selected proteins with one or more ubiquitin molecules. These ubiquitinated proteins are then recognized and sucked into the proteasome by proteins in the stopper. Proteins that are meant to be short-lived often sport a short amino acid sequence that identifies the protein as one to be ubiquitinated and ultimately degraded by the proteasome. Denatured or misfolded proteins, as well as proteins containing oxidized or otherwise abnormal amino acids, are also recognized and degraded by this ubiquitin-dependent proteolytic system. The enzymes that add ubiquitin to such proteins recognize signals that become exposed on these proteins as a result of the misfolding or chemical damage. For example, amino acid sequences or conformational motifs that are normally buried and inaccessible. There are many steps between DNA and protein. We have seen so far in this chapter that many different types of chemical reactions are required to produce a protein from the information contained in a gene. Figure 740. The final concentration of a protein in a cell therefore depends on the efficiency with which each of the many steps is carried out. In addition, many proteins, once they leave the ribosome, require further attention before they are useful to the cell. For example, covalent modification, such as phosphorylation, the binding of small molecule cofactors, or association with other protein subunits are often needed to activate a newly synthesized protein. Figure 741. We will see in the next chapter that cells have the ability to change the concentrations of most of their proteins according to their needs. In principle, all of the steps in Figure 740 can be regulated by the cell. However, as we shall see in the next chapter, the initiation of transcription is the most common point for a cell to regulate the expression of each of its genes. Transcription and translation are universal processes that lie at the heart of life. However, when scientists came to consider how the flow of information from DNA to protein might have originated, they came to some unexpected conclusions. RNA and the origins of life. To understand fully the processes occurring in present-day cells, we need to consider how cells evolved. But one key process, that of gene expression, presents a particular challenge. If nucleic acids are required to direct the synthesis of proteins, and proteins are required to synthesize nucleic acids, how could this system of interdependent components have arisen? One view is that an RNA world existed on Earth before modern cells appeared. Figure 742. According to this hypothesis, RNA, which today serves as an intermediate between genes and proteins, both stored genetic information and catalyzed chemical reactions in primitive cells. Only later in evolutionary time did DNA take over as a genetic material and proteins become the major catalysts and structural components of cells. If this idea is correct, then the translation out of the RNA world was never completed. As we have seen in this chapter, RNA still catalyzes several fundamental reactions in modern cells. These RNA catalysts include the ribosome and RNA splicing machinery, can thus be viewed as molecular fossils of an earlier world. Life requires autocatalysis. The origin of life requires molecules that possess, if only to a small extent, one crucial property: the ability to catalyze reactions that lead, directly or indirectly, to the production of more molecules like themselves. Catalysts with this special self-promoting property, once they had arisen by chance, would reproduce themselves and would therefore divert raw materials from the production of other substances. In this way, one can envisage the gradual development of an increasingly complex chemical system of organic monomers and polymers that function together to generate more molecules of the same type, fueled by a supply of simple raw materials in the environment. Such an autocatalytic system would have many of the properties we think of as characteristic of living matter. The system would contain a far from random selection of interacting molecules. It would tend to reproduce itself. It would compete with other systems dependent on the same raw materials. And, if deprived of its raw materials or maintained at a temperature that upset the balance of reaction rates, it would decay toward chemical equilibrium and die. But what molecules could have had such autocatalytic properties? In present-day living cells, the most versatile catalysts are proteins, which are able to adopt diverse three-dimensional forms that bristle with chemically reactive sites. However, there is no known way in which a protein can reproduce itself directly. Nucleic acids, however, can do both of these things. RNA can both store information and catalyze chemical reactions. We have seen that complementary base pairing enables one nucleic acid to act as a template for the formation of another. Thus, a single strand of RNA or DNA can specify the sequence of a complementary polynucleotide, which in turn can specify the sequence of the original molecule, allowing the original nucleic acid to be replicated. Figure 7.4.43. Such complementary templating mechanisms lie at the heart of DNA replication and transcription in modern-day cells. But the efficient synthesis of polynucleotides by such complementary templating mechanisms also requires catalysts to promote the polymerization reaction. Without catalysts, polymer formation is slow, error-prone, and inefficient. Today, nucleotide polymerization is rapidly catalyzed by protein enzymes, such as the DNA and RNA polymerases. But how could this reaction be catalyzed before proteins with the appropriate catalytic abilities existed? The beginnings of an answer to this question were obtained in 1982, when it was discovered that RNA molecules themselves can act as catalysts. We have already seen in this chapter, for example, that a molecule of RNA catalyzes the peptidyltransferase reaction that takes place on the ribosome. The unique potential of RNA molecules to act both as information carriers and as catalysts is thought to have enabled them to play the central role in the origin of life. In present-day cells, RNA is synthesized as a single-stranded molecule, and we have seen that complementary base pairing can occur between nucleotides in the same chain. See Figure 7.5. This base pairing, along with non-conventional hydrogen bonds, can cause each RNA molecule to fold up in a unique way that is determined by its nucleotide sequence. Such associations produce complex three-dimensional patterns of folding, where the molecule as a whole adopts a unique shape. As we saw in Chapter 4, protein enzymes are able to catalyze biochemical reactions because they have surfaces with unique contours and chemical properties on which a given substrate can react. In the same way, RNA molecules with their unique folded shapes can serve as enzymes, Figure 7.44, although the fact that they are constructed of only 4 different subunits limits their catalytic efficiency and the range of chemical reactions compared with proteins. Nonetheless, ribozymes can catalyze many types of chemical reactions. Most of the ribozymes that have been studied were constructed in the laboratory. Table 7.4. Relatively few catalytic RNAs exist in present-day cells, but the processes in which catalytic RNAs still seem to have major roles include some of the most fundamental steps in the expression of genetic information, especially those steps where RNA molecules themselves are spliced or translated into protein. RNA, therefore, has all the properties required of a molecule that could catalyze its own synthesis. Figure 7.45. Although self-replicating systems of RNA molecules have not been found in nature, scientists appear to be well on the way to constructing them in the laboratory. Although this demonstration would not prove that self-replicating RNA molecules were essential in the origin of life on Earth, it would demonstrate that such a scenario is possible. RNA is thought to predate DNA in evolution. The first cells on Earth would presumably have been much less complex and less efficient in reproducing themselves than even the simplest present-day cells. They would have consisted of little more than a simple membrane enclosing a set of self-replicating molecules and a few other components required to provide the materials and energy for their replication. If the evolutionary speculations about RNA outlined above are correct, these earliest cells would also have differed fundamentally from the cells we know today in having their hereditary information stored in RNA rather than DNA. Evidence that RNA arose before DNA in evolution can be found in the chemical differences between them. Ribose, see Figure 7.3a, like glucose and other simple carbohydrates, is readily formed from formaldehyde, HCHO, which is one of the principal products of experiments simulating conditions on the primitive Earth. The sugar deoxyribose is harder to make, and in present-day cells, it is produced from ribose in a reaction catalyzed by a protein enzyme, suggesting that ribose predates deoxyribose in cells. Presumably, DNA appeared on the scene later, and then proved more suited than RNA as a permanent repository of genetic information. In particular, the deoxyribose in its sugar-phosphate backbone makes chains of DNA chemically more stable than chains of RNA, so that greater lengths of DNA can be maintained without breakage. The other differences between RNA and DNA, the double helical structure of DNA and the use of thymine rather than uracil, further enhance DNA stability by making the molecule easier to repair. We saw in Chapter 6 that a damaged nucleotide on one strand of the double helix can be repaired by using the other strand as a template. Furthermore, Deamination, one of the most common unwanted chemical changes occurring in polynucleotides, is easier to detect and repair in DNA than in RNA. See Figure 623. This is because the product of the deamination of cytosine is, by chance, uracil, which already exists in RNA, so that such damage would be impossible for repair enzymes to detect in an RNA molecule. However, in DNA, which has thymine rather than uracil, any uracil produced by the accidental deamination of cytosine is easily detected and repaired. Taken together, the evidence we have discussed points to the idea that RNA, having both genetic and catalytic properties, preceded DNA in evolution. As cells more closely resembling present-day cells appeared, it is believed that many of the functions originally performed by RNA were taken over by molecules more specifically fitted to the tasks required. Eventually, DNA took over the primary genetic function, and proteins became the major catalysts, while RNA remained primarily as the intermediary between connecting the two. Figure 746. With the advent of DNA, cells were able to become more complex, for they could then carry and transmit more genetic information than could be stably maintained in an RNA molecule. Because of the greater chemical complexity of proteins and the variety of chemical reactions they can catalyze, the shift, albeit incomplete, from RNA to proteins also provided a much richer source of structural components and enzymes. This enabled cells to evolve the great diversity of structure and function that we see in life today. Essential concepts. The flow of genetic information in all living cells is DNA to RNA to protein. The conversion of the genetic instructions in DNA into RNAs and proteins is termed gene expression. To express the genetic information carried in DNA, the nucleotide sequence of a gene is first transcribed into RNA. Transcription is catalyzed by the enzyme RNA polymerase. Nucleotide sequences in a DNA molecule indicate to the RNA polymerase where to start and stop transcribing.